Okay, hello everybody. I'm Ken Waterman, the president of FreeThink Technologies, and I'm very pleased to welcome everybody to the first of this new webinar series. Today, I'm going to be introducing our speaker in a second, and I will tell you a little bit about things. First, let me tell you, FreeThink is hosting this, and just couple lines about who FreeThink is. So for those who aren't used to us, we both produce the software ASAP Prime and carry out laboratory work for very fast and accurate accelerated stability determination, shelf life determination. And we also develop drug delivery technology and use drug delivery to solve complex formulation challenges. And we've just launched our new technology, OsmoCap, which is an osmotic oral controlled release capsule. And we do work in other fields of solubility technologies, depots, and other technologies. Today's speaker for our new series on nitrosamines or N-nitrosamines in drug products is Maria Ultramari. Dr. Ultramari is the CEO of Spectra Consultancy, which focuses on impurity assessments for compliance with ANVISA and ICH and represents FreeThink in Latin America. She is a pharmacist and toxicologist by training. She's a member of the board of directors of the Brazilian Society of Toxicology. She has a lot of experience with scientific and regulatory aspects of analytical development and the toxicology of impurities, where she's especially adept at employing in silico tools to assess integrated risk. Dr. Ultramari will be speaking to us about new approaches to setting acceptable intake levels for N-nitrosamines. People who have questions, you can feel free to enter the questions, type in the questions in the Q&A box that everybody should have at any time. I will be, at the end, reading selected questions out, and all the questions will be captured and attempt to answer if we don't get to all of them. This session will be recorded and will be available after a day or two from the FreeThink website. And be aware also all the past webinars that we've had are available all free for everybody. And with that, I'm going to turn over control to Maria and let her take it from there.
Thank you, Ken. Hi, everybody. I don't know if it's good morning or good evening for you. I don't know where you are. We are from all over the world today. I will share my screen here. Just let me project. Okay. Can you see my screen?
Yes, we can, Maria. Yeah, we can see it. Yeah.
Thank you, Rachel. So, thank you very much for this opportunity. It's a huge pleasure to introduce this webinar series that is promising because we have many the greatest scientists in this field coming in the next series, in the next webinar. So today I will talk to you about let me just put here. Oh, thank you, my question. The new approaches to set acceptable intakes levels for nitrosamines drug substance related impurities. So it's the complex nitrosamines that are pretty much the focus today in the investigation of nitrosamines in pharmaceutical products. As Ken kindly introduced me, I'm Maria Ultramari. I am a toxicologist and CEO from Spectra, and I am a Brazilian, and I will represent here not only Brazil, but Latin America to discuss this topic. So thank you for this opportunity, FreeThink. It's a huge pleasure. To start with this, the goal of this presentation today is to share with you all the new aspects that are being discussed regarding the definition of the acceptable intakes for nitrosamines. And this presentation was based on the most recent papers published in the literature of the greatest scientists that we have publishing. And I would like to thank all the scientists that are publishing all the information regarding nitrosamines, from the risk assessment to the acceptable intakes determinations, because this is the way that the science is improving on this theme. It's very, very important, not only for the whole world, but especially for the generic companies that I will give some highlights by the end of this presentation today. So, and I will talk to you about the historic content, focus on the acceptable intake, a definition of nitrosamines, the challenges to set nitrosamine drug substance-related impurities acceptable intakes, new approaches to set the acceptable intakes that was published by EMA and FDA very recently, and what we think it's the next steps. And by the end, I will bring you some information regarding the generic companies. So we all already know about the history of the nitrosamines, but just to give you an introduction of this theme, and also to do this kickoff of these webinars to deliver to my colleagues that comes next. I would like to give this background, saying that NDMA was the first nitrosamine discovered and found in valsartan in 2018, and it's been 5 years that we've been discussing this whole problematic regarding nitrosamines. We all know that NDMA, it's a known mutagen and a potent carcinogen in animal studies. And because all of that, the health authorities, they made recalls, and the first recall that they did was the valsartan and all the pharmaceutical products that contained valsartans. And after that, we have many recalls, for example, the ranitidine, ibezapine, metformin, and many others. And with that, the health authorities, they started to publishing the guidelines to mitigate the presence of the nitrosamine in pharmaceuticals. And this is pretty much interesting, because this is this thing it's developing so fast that all the guidelines are being reviewed in a very, very fast way. So we need to be very alert and following the literature, and also the guidelines and the EMA and especially FDA to follow all these improvements and reviewers that they are doing. Regarding the nitrosamines, we already knew about them. They are described in ICHM 7 as a court of concern group. So this means that they are a very special group, a very potent carcinogens and mutagens. And this means that the TTC, the threshold of toxicological concern described in ICHM 7, doesn't apply to control those nitrosamines. So that's why the health authorities are providing acceptable intakes for nitrosamines. And from the very beginning, they started providing limits for the low molecular weight nitrosamines. And here we can find the most relevant and what is being published about the low molecular weight nitrosamines. It means that we have a limit for NDMA of 96 nanograms per day, and DEA, that is the most potent one, it's 26.5 nanograms per day, and all the others low molecular weight nitrosamines have this acceptable intake defined by the regulatory agencies with different methods. And they've been using the linear extrapolation from TD50 as described in ICHM 7, and also they are using the approach that we know as read across. So if we go back to these limits, acceptable limits defined by the regulatory agencies, we will see that they repeat for different molecules, and this means that they are using some techniques like read across to establish these limits for, especially for the nitrosamines that we don't have carcinogenic studies published, or they are not, if they are not robust enough to set the limit for or to define a TD50. And this approach that the health authorities are using has some gaps, because they are not really transparent and not comprehensive, because for each case they use different approaches. So this means that we need to get deep into these concepts and find the studies that they are using to provide all these acceptable limits. And for sure the scientist community, it's looking for new evidence and trying to better understand from where they took those informations to set limits. And then another time point that is very, very important during the nitrosamines discussions and assessments is when we had in 2021 the recall of Chantix, that it's the varenicline, I'm sorry, that was the first nitrosamine drug substance related impurity. And from that one, we are pretty much focused on those nitrosamines, those complex nitrosamines, mainly because we don't have many studies or information to set acceptable limits for them. And this was followed by recalls and discovering another nitrosamines drug substance related in propranolol, enalapril, and phenadrine, and so on. And the most problematic thing that we have nowadays is the lack of the acceptable intakes for those complex nitrosamines. And because of that, we have many challenges to set the acceptable intakes for the drug substance related impurity nitrosamines. We need to pay attention because they are very different. They have different chemical spaces. When we compare these molecules to the low molecular weight molecules, that those low molecular weight molecules have data, carcinogenicity studies data available, and this is not the case of the nitrosamine drug substance related impurities. We need to pay attention always on the amine, on the alpha carbonamine, because they need to suffer high alpha hydroxylation. That is the key metabolic pathway to transform this nitrosamine into a very potent carcinogen. And as we don't have carcinogenicity studies available, we need to bring many different approaches to set limits for those nitrosamines and guarantee that the patients are not are safe. And we've been discussing, and EMA and FDA are publishing many different approaches to define limits for those nitrosamines. And the first approach that they published was define a TTC of 90 nanograms per day for those nitrosamines that we don't know anything about it. We don't have studies. And the first thing that they did was setting this limit. And also, as this limit, it's a very conservative limit, they allowed us to use an interim limit of 178 nanograms per day only for 1 year during the investigations and all the controls that we need to do when we first discover a real nitrosamine drug substance related, or any nitrosamine that doesn't have any acceptable limit established. And when we EMA published this TTC, the whole scientific community was shocked, because it's a very, very impact and conservative limit, and this impacts a lot in the method development and all the approaches that we need to do after doing the risk assessment, and to find the concentration and of those nitrosamines in the drug substance or drug product. And the challenge is that, and the most, we say that these values are very conservative, because it was set based on low molecular weight nitrosamines. So if you go and if you see the low molecular weight nitrosamines and you compare to these non-nitrosamine drug substance related impurities, they have many differences. So, and as I told you previously, we have different chemical spaces, and so we need to pay attention and respect all these differences to better understand if we really have a substance that we need to I don't know, to set the limit and to be safe, and if compare them if they really is a potent carcinogen and mutagen. And this other interim limit set only for 1 year, it's not sufficient also, because many controls and improvements that we need to do in the processes or in the route of synthesis, or even in introducing new controls in the pharmaceuticals, it's not done in only 1 year. So, and after that, after using this interim limit, we also need to establish a proper limit to the nitrosamine that we want to control in the pharmaceutical product. So it was something that helped, but we know that nowadays it's not helping anymore. And following all those discussions and all the guidelines published by the EMA and FDA, especially regarding this TTC that is very, very conservative, many publications were available and in the literature, and one of them was this from Crystal Dobo, that she could classify and different nitrosamines and give them special acceptable intakes according to special features. So she could separate the nitrosamine groups into 13 different groups with different acceptable intakes. And from this paper, we could now get deep into the read across and structure activity relationships to better understand what acceptable intake are we going to consider for the molecules, especially the known nitrosamine drug substance related impurity. We can see here that we still have the 90 nanograms per day. This happens when we don't have studies or especially carcinogenic studies, carcinogenicity studies to set limits better than these 18 nanograms. But most of the other classes, we could set higher limits, much higher limits than this 18 nanograms. And from this paper, as I told you, we could get deep into the read across, because we could look at all those features and all those nitrosamines and known nitrosamines with different studies and information to set limits, to extrapolate those information to the nitrosamines that we are interested in. So, and this opened this whole concept of using the read across and SAR strategies to set limits for nitrosamines drug substance related impurities. But we still have many challenges, because for us to select a surrogate compound to do the read across evaluation, we need to look at similar structurally around the nitrosamine substructure with similar substitution patterns. So this is not easy, because we also need that the surrogates have robust carcinogenicity data, similar metabolism, because we need the activation for these nitrosamines to become a real potent carcinogen, and also look at similar drug metabolism and pharmacokinetics. Those informations are very important for us to set and to define a good read across to set limits for unknown nitrosamines. And this is a huge challenge, because it's very hard to find all those informations to use surrogate properly to set limits. And because of this complex evaluation, EMA and FDA set new approaches to set limits for those nitrosamines, especially this complex nitrosamines, and they published very recently the CPCA approach, that it's a carcinogenic potency categorization approach. It's pretty much based on SAR evaluation, because they are looking at the molecule and all the features and patterns that we need to focus on to establish some limits. But this is not only the approach that they are suggesting in this revision of the Q&As that was recently published. We also can use the TD50 from surrogates as a point of departure to do the read acrosses or SARs. We also can use the modified Ames test to prove the mutagenicity of the nitrosamines, but here we have a very complex situation, because having a negative result in the Ames tests do not allow us to consider this as a non-mutagenic impurities. So if we have negative results in this modified Ames test that EMA and FDA are suggesting, we need to control these nitrosamines as a mutagen at the TTC of 1.5 micrograms per day. This is pretty much different from what ICH-M7 allows us to do, because if we prove by the Ames test that this is not a mutagen, we can go to a class 5 impurities and control those impurities as non-mutagenic by using the ICH Q3A or Q3B, but this is not the case here for nitrosamine, not yet. So EMA is also, and FDA they are also suggesting to conduct relevant and well-conducted in vivo studies to prove that this molecule is negative, and only with that we are allowed to control those nitrosamines as non-mutagenic impurities. And considering this CPCA approach, the CPCA in fact considered this hydrogen on its alpha carbons, and also if the molecule has deactivating and activating features. And if we compare, for example, MDMA, then we know that it's a highly potent carcinogen, and this alpha carbon hydrogens available for metabolism. And if we compare to, for example, N-nitroso hydrochlorothiazide, we can see that the alpha carbon and alpha hydrogens are not completely available. Here we have only 2 hydrogens available for metabolism. If we see the vanericline nitroso, we have at both sides 2 hydrogen available for metabolism. And for example, in nitroso-sertraline, we have 1 here and also 3 hydrogens here. And this is pretty much the approach, and also considering the deactivating and activating features of those molecules, we can set some scores and then go into this classification of the nitrosamines. So EMA and FDA, they also did a classification for those nitrosamines, considering all this information that I said previously. So if the molecule have any hydrogen on its alpha carbons, if there is any, we can consider it as a class 5 and control as using the TTC of ICHM7 and so on. And the less if the alpha carbon hasn't hydrogens available to metabolism, we can consider it as category 5, and then we will do this assessment of each molecule to see if it has deactivating or activating features, and then we will do some scores that are presented in the Q&A from EMA and FDA, and to set the categorization of those molecules. And with that, we can come to this values, the acceptable limits for each classification. And if we can see here that for class 5 and class 4, we can control those nitrosamines in TTC using TTC of ICHM7. And if it's class 3, we can use the 400 nanograms per day, class 2, 100 nanograms per day. And if it's class 1, we still need to control using the TTC that they provided for nitrosamines. So complex nitrosamines, that it's 18. And this is still very, very challenging. And as EMA, FDA also did this CP, recommend the CP CA, but the difference is that here they are not considering the TTC for nitrosamines. They are considering the acceptable limit for MDEA, and it is improvement, but not sufficient at all for many different molecules. And also a new approach that are being discussing a lot in the scientific community, it's conducting these in vivo mutagenic tests. And for sure that transgenic mutagenic mutation in rodent, it's the most acceptable test, in vivo test, the most acceptable from the regulatory perspective. But other in vivo mutagenic tests are being discussing in the literature. This very new duplex sequencing, it's a very promising technology and strategy to control and to assess the mutagenicity, not only the mutagenicity of the molecules. And we can also use the COMET assay, that it's a very, very well-known protocol to conduct mutagenic assessment. And we have a very, very recent publication comparing these 3 methods to these 3 in vivo methods using NDEA. And the conclusion of this paper written by Berko and colleagues, is that for sure the transgenic in rodent, it's the most acceptable test for the assessment of mutagenicity, especially in nitrosamines, but it is a very limited availability of these transgenic animals to conduct this study, and also we do not have many qualified laboratories to conduct these studies, and this is a very, very challenging situation, especially when we are thinking about the countries that and the countries that are not are most working with generics. Here in Brazil, for example, we do not have any, not that I know, any laboratory that is conducting this kind of test. But we also have this duplex sequencing being discussed and having more and more data published, that it's very comparable, as this paper said and brought, this very comparable to this most acceptable test. It uses non-transgenic animals, and we have faster results with this method. So because of that, it's a very promising technology, but may not be accessible to everyone, not yet. So, and a very nice thing of this test is this new technology, is that we can have the mechanistic information, so we can see the overall toxicological understanding. We can have the overall toxicological understanding, not only the mutagenicity. And also we can have the common assay, and as the paper said, it's a very well-known and sensitive method to detect this mutagenicity for nitrosamines. It also doesn't use a non-mutagenic animals, and it's a faster result, and it's comparable to this transgenic in rodent. So those technology, those tests are very promising, and they are being using to evaluate the nitrosamines and to help to set limits, because if they are very well designed, we can set limits from acceptable intakes from those all those tests, in vivo tests. But all those approaches that EMA and FDA are provided in the last Q&A revision, they also have gaps. So we need to consider different molecular weight, because at this point we are not considering these differences, and also regarding the lasting lifetime approach, they are saying in the Q&A that it is a temporary measure during the evaluation and the CAPA implementation, but the scientific community is working hard to prove that less than lifetime approach, it is protective and can be acceptable, even if we think of this concept, if we consider that NDA and DEA, the most potent nitrosamines that we know, was considered to set this approach in the ICH 7. This could be a very nice alternative to set limits or real limits to the nitrosamine drug substance related impurities. And also the positive results in vivo and in vivo tests now from the TGR, for example, do not define acceptable intakes. So we are, the scientific community are working on that to design those tests to have acceptable limits defined by those tests. And considering all of that, We have some initiatives happening in the world. So the HESI group, or even LASA, or FPIA are working hard to set all those acceptable limits and to prove that those methods are protective and they are pretty much giving us confident results for us to set those limits and prove that these nitrosamines, especially these complex nitrosamines, are not potent carcinogens. And next steps, it would be very, very nice if we could prove that the Ames test is sufficient to prove mutagenicity of nitrosamines, and with that we could have negative results and consequently controlling these non-mutagenic impurities as the guidelines Q3A and B. And well-designed in vivo studies could give us benchmark doses for us to set the acceptable intake limits. And also it would be very, very nice, again, if we could use the less than lifetime approach to support the acceptable intake determination. And the last thing that I want to show you and to share with you, it's how we are conducting all those assessments when we think about generic companies. So from a generic company's perspective, as Brazil has most of our pharmaceutical companies are generic, and also in Latin America, we consider a very huge investment to conduct all those experiments. So here the mindset is always invest or withdraw the product from the market, and this has a huge impact in the public health when the companies are considering this withdraw or stop producing the products that they are investigating on nitrosamines and so on. So that's why we depend a lot on scientific publications. So I really encourage all those scientists that are doing a brilliant job to publish more and more, because only with that we can do our risk assessment properly and really identify the risk, the real risk on our products here. Nowadays, here in Brazil, we are still in phase 1. We are doing our risk assessments aligned with the new approaches. So the good thing of being late on those assessments is that we are following everything that are being publishing in the literature. So our risk assessment are pretty much focused on nitrosamine drug substance related impurities, not only the low molecular weight nitrosamines. So I can tell you that our risk assessments are very, very robust, and given that we have all those informations available by this time. Anvisa is following the EMA and FDA approaches. I can tell you that from now, Anvisa is receiving all these first risk assessments that we are conducting here in Brazil. So we don't know yet how Anvisa is going to receive and understand all those approaches that we are using that are aligned with the international community. But let's see, this is another chapter of our life here in Brazil. And what I can tell you from the other countries in Latin America is that now they are starting only now the risk assessment of nitrosamine. So we still don't know what we are going to have from a risk assessment or risk point of view. And with that, I want to thank all my team here from Spectra that are working very hard to follow all those publications that are being publishing every month. And we are doing a very nice job, and I know that they are watching here. So thank you guys Without you, this won't be possible. And thank you very much for this opportunity, and I'm open for any questions.
Thank you, Maria. It's very good. And we do have a couple of questions, and I want people to start typing in questions that will give her some challenge. This field has become, from, as you said, just a few years ago, who even thought about nitrosamines, to withdrawing of some products. I just was curious, from a toxicological standpoint, one of the drugs you mentioned that was withdrawn was Chantix or varenicline, which is a drug that has been very effective at getting people to stop smoking.
Yeah,
I'm
I know
curious,
what you're going to say.
the public health risk as part of the assessment, if someone's taking varenicline and actually stops smoking successfully, is the risk from N-nitrosamines at these extremely low levels greater than the risk of continuing to smoke?
Yeah, yes, this is crazy. Again, I agree with you. It's the pharmaceutical industries, they are doing a huge efforts to control nitrosamines, and when we see the tobacco or even the food that we know that have many nitrosamines available, not yet they are not being controlled as we are controlling the pharmaceutical. So this is we know that the regulatory agencies are focused on foods too, and also cosmetics, and many types of products are being investigated from the nitrosamines perspective. But this is very curious because tobacco has a lot of nitrosamines available. So why are you,
among other carcinogens,
worried? Yes.
All right, so let me give you a couple of questions that have been typed in, and again, encourage people to put in questions. Is it allowed to perform a standard Ames test and then a modified Ames test, confirmatory, if negative results are found on the standard test?
Yes, Tanya. In fact, I don't have any experience on that, but thinking about the what are being published, we know that the regular Ames tests are not being acceptable for EMA and FDA because there are some complexities on the S9 used and the solvents that are being used and pre-incubation and so on. So I haven't seen any work, any paper comparing the test, Ames test, the regular, the standard Ames test with the modified tests. I know that this is a proven concept. That's why EMA and FDA are encouraging us to run the modified tests. But the fact is that for nitrosamines, even Ames tests, we just use that to consider a regular mutagen and not court of concern mutagen.
Yeah, that makes sense. Next question. There are a number of nitrosamines on the EMA's list that didn't make it on the FDA's list. Any idea why or what to do about that?
Yes, I think FDA did a good job by having many molecules assessed and with the acceptable intake defined. They use the CPS CA. So if you do the assessment using the CPCA, you will come to the same results as FDA. So I don't have any idea. Maybe they have many people or more time to calculate all those limits.
Okay. Is there a different approach with oncology products for use of S9 for impurities? I'm assuming the question is because with oncology, you often are given more relaxed limits because of the nature of.
Yes, this is another, yes, for oncology products, especially for cytotoxic product, SHM7 doesn't apply. So it's a different concept. So we are allowed to have mutagenic impurities on those drugs. So it's a different approach. Yeah.
I mean, one of the bottom lines is this is all a new frontier.
Yes.
And
Yeah.
everybody's still trying to figure it out. But I think what you're saying, which I think people concur to, is well, who knows what each health authority will say. You'll probably get an easier time with respect to oncological products.
We hope so.
We hope so, yes.
Yeah.
Are there any computational software or tools available to screen or to do risk assessments with respect to the toxicology? We're going to be hearing, I want to make people aware, we're going to be hearing a talk about risk assessment software with respect to chemical structures, not to do with necessarily toxicity, but to do with what they will generate. But in terms of toxicity, anything available?
Yes, in fact, for risk assessment, I haven't seen anything. I know that the risk assessment you have to do looking at all those parts of the DMF and all the processes in your drug product processes and so on. So I haven't heard about anything yet. But regarding the toxicity, we have the established software like LASA, Derek, and Sarah, and so on. But at this time, as we need to run many different tests, especially in vivo tests, we do not have anything, not from that I know, okay, ready to use. Yeah.
How do you apply the CPCA approach for tertiary amines that afford and nitrosamines, NDSRI? Tertiary.
Yes, tertiary means that, in fact, if you follow the CPCA, it's very explained. They put many different exercises for you to follow. I don't know yet how to answer the discussion, because I have a whole team here wants to that look at the chemical structures, and I do the risk assessment with the toxicity. But Isabel, I don't know if you're from Brazil or Latin America. If you want me to get in touch, we can ask this later.
All right, and then another question
Answer
was,
this later.
that's fine. Can you kind of, in your opinion, when is the right time in terms of clinical development phases to do a formal risk assessment?
Oh my
I mean,
God.
just to be clear, this is tricky. I'm going to help you here because you may not need to do phase 1, but if you like your product, what are you going to do later? And that's where we at FreeThink get involved a lot, because now you have a product, you want to go forward. How do you know whether it's good or not? How do you know if you fixed it? So
yes, Ken, I don't know. I have the experience here in Brazil of Nathan, for example, my partner here, that was developing new drug substance. So we were pretty much focused on, at that point, we were pretty much focused on the synthesis, if you are using the right reagents and if you are using the right solvents, not to have any problem in the future. For sure, the synthesis is only one thing that we need to focus on, but then the processes, and also the excipients that are being discussed a lot because of the nitrates contaminations and so on. So in fact, we need to look at that. And I think that this topic, this nitrosamines, brought us a new mindset for us to get into deep into what we are doing, So it's not, and it's not only about nitrosamines. I think we need to pay attention on all the mutagenic impurities that could be formed during all those processes. I hope nitrosamines, it's the first one and the last one, but we know that this is not true. Yeah.
Part of the reason this is so challenging, especially for drug product, is that you're also talking about trying to develop analytical methods. And
Yeah.
normally our analytical methods are not designed to see things at the parts per billion
Yeah.
level. And
Yes.
we would ignore this in drug product stability in many cases. So
Yes.
this
and
is
that's
part of the knowledge.
And that's why the TTC of 99 grams per day, it's so challenging. It's very hard to accomplish that. Yeah.
Okay, I believe USP intends to provide tests, guidances for nitrosamine contamination by 2025. Do you know anything about this?
Yes, in fact, I know that USP are developing a chapter on that. I don't know, in fact, what they are going to publish, but probably regarding to the developing the method, the analytical methods. I don't know if they are going to give any information regarding risk assessment, but because they are pretty much focused on methods.
Yep. Okay. If a surrogate nitrosamine with robust data has been identified, should the limit be obtained by CPCA? Is that valid or not? I think the question in some ways is how good is it to use surrogates? Let's ask it that way.
I think it's pretty much confident when the surrogate has robust data, really. So we are doing that not only for nitrosamines, we have been doing that for any impurities, qualification of impurities. So the read across, it's a very protective approach for us to set acceptable intakes. And I don't think that you can compare the approaches that using the surrogate and do a read across and the limits that the CPCA will give you. Probably, I don't know which molecules are we talking about, but probably CPCA will be more conservative than a good read across with the surrogate.
Is the categorization approach, which I assume means the read across, alone sufficient to set the limits, or do we need to also look into other options listed in the guidances and enhanced aims, TD50, of surrogate and other things?
Yeah, if you have this information, you should use it. So the TD50, if you have the and also there's something that is very important to clarify here too. If you have the results of carcinogenicity studies, it doesn't mean that you don't need to look at those studies or search in the literature more about those molecules, because there are some papers that are discussing different approaches, even using the same database and information of that EMA and FDA used. So there are different ways to look at those studies, and they are if they are robust, if they were run in GLP approaches. So it's if you compare, the best thing is to have a carcinogenicity study. If you don't have, you can do a read across or a CPCA. I don't know exactly, because I think there will be many different molecules, but probably CPCA will be more conservative. And if you have a surrogate with all those information, and please pay attention on the metabolism, on the solubility of those molecules, and the administration route, because sometimes this nitrosamines is not being bioavailable to interact with the receptors, or even being metabolized to become a potent carcinogen. So all those informations you need to consider to set limits, because CPCA, it's only looking at the molecules and the structures and the alpha carbon hydrogens and the activating and activating. So if you have more data, if you have surrogates that that has all those data for sure. It would be better for you to set limits based on that.
Okay, well, I think we're coming to the end of our time, and anybody who has further questions can certainly bring them directly to Maria at her website, or if they have trouble, they can go through FreeThink's website. We'll get back.
Yes.
And there are some people requesting things. And again, there will be a recording of this available the next few days. Give us a week or something to get it up. Just sign in through the FreeThink website. You can download this webinar and the entire series. The next one in our series, which will be 2 weeks from now, will be on featuring, let's see if I can put up the image. We're featuring Dr. Rachel Hemingway about use of an in silica tool to determine the molecular susceptibility of compounds with respect to forming nitrosamine degradation products. So that'll be kind of a nice complement to this. Here you'll learn, well, what's the risk? Well, what's the risk of forming such compounds? I think that'll be very exciting. So I'll be in 2 weeks. You can register for that if you've not already, and we look forward to more people. And with that, we'll give a virtual applause to our speaker. And thank you again. Bye, everyone.
Thank you. Thank you, guys. Thank you, Ken. Bye.
Bye.